So, in photosynthesis, we again were focusing on the equation for this particular series of reactions, which you'll recall is carbon dioxide plus water plus energy in the form of light, which yields glucose or some form of simple sugar and molecular oxygen. Now, whereas we focused on the gases in respiration, here in photosynthesis, what we're going to focus on are two things. We're going to focus on the absorbance of light and the pigments that are involved in that absorbance. And we're going to focus on what the cells do in plants when they make an overabundance of this sugar. And so those are the two things we're primarily going to focus on. And to focus on the light, what we really need to focus on initially are these specialized molecules called pigments that can ultimately absorb that light energy. And so the pigments in question, there are technically four. There is chlorophyll A, which you may recall is the primary photosynthetic pigment in plant, meaning it's the primary pigment that's utilized in photosynthesis. Chlorophyll B, which also expands the absorbed spectrum of light. And then we have these other two called xanthophyll and carotene. And these two, the xanthophyll and carotene, are commonly referred to as carotenoids. So those are carotenoids. So in your first exercise, what you tried to determine was where in the visible spectrum of light these pigments could absorb light the best. And this particular spectrum that you devised in lab should pretty closely match what you observed in the book. Okay, so what you did there is you took two different extracts, carrot extract and spinach extract. You took spinach extract. Somebody tell me, which of these pigments do you think are in spinach? So chlorophyll A and B, but guess what else is there? Xanthophyll and carotene. Spinach extract, as you later found when you rubbed the spinach leaf on the chromatography paper, spinach has all of these pigments. Okay? So spinach extract has all four pigments. But you also used carrot extract. Which pigments do you think are going to be in carrot? Just the carotenoids, right? Good. Now, just so you understand, this is going to be pretty much what you'll see in your book. If you go into your book on the chapter on photosynthesis, you should see something that looks relatively close to this. So I'm going to draw my wavelengths here from 400 to 700 nanometers. And then here's going to be my absorbance spectrum. So this is 1 up here, this is 0. Okay. So, chlorophyll A has a peak that's pretty close to the purple-blue threshold before falling off here, and then it has a secondary peak right over here around the red and orange threshold. So this is going to be chlorophyll A. Primary photosynthetic pigment in plants. Notice its high point is pretty darn close here to blue and purple. Somebody want to remind me? Purple light. High energy or low energy in terms of the energy associated with it? High levels of energy. It would make sense for photosynthesis if we're trying to capture a lot of light energy, we want to capture the light with the most energy. Okay? Chlorophyll B is going to have a peak that's a little higher than A's, but it's been shifted a little bit, okay, in regards to where it is. Chlorophyll B predominantly absorbs light best in the blue range of the visible spectrum. So the blue line here is going to be B. Now real quick, you'll notice that both A and B tapered off right around this region. What color corresponds to this region of the visible spectrum? Green. Green is not that well absorbed by these photosynthetic pigments. All right, now finally, the carotenoids are support or accessory pigments. They absorb excess light that could otherwise damage our two chlorophylls. Now tell me, those chlorophylls, what color light is the most dangerous to them? Light that's found over here in the red-orange part of the spectrum, or light over here in the blue-violet part of the spectrum? Blue and violet. Y'all just told me blue and violet have the highest level of energy. That's going to be the most dangerous in terms of damage. There's a lot of energy there, it can do damage. The red and orange, that's low energy. It's not going to have that much tendency to cause damage. So where do I expect to see my carotenoids absorb the most light if they're trying to absorb light to protect the chlorophylls? Blue and violet, and that's exactly what we see. We see something like this for the carotenoids, and then they completely drop off over here in the red-orange. So red over here is going to be carotenoids. Okay, now let's go back to what you actually observed in lab. So, one of your extracts has all 4 pigments in it. The other only has the carotenoids. So let's do the easy one of these two first. What should your graph for carrot extract, what should it have looked like? Well, if I only have the carotenoids, what do I expect to then see when I put this sample into the spectrophotometer in terms of absorbance? Just the graph I've drawn here for the carotenoids. Does everybody get that? If all that's in that carrot extract are the carotenoids, then my absorbance spectrum should be almost exactly what I have drawn here. That make sense? Okay. Now for spinach, however, it contains all 4 pigments. So, what should its absorbance spectrum look like? Should it look like any one of these? Not one, but what should it look like instead? All of them together, right? And that's most likely what you all saw, was you probably saw your spinach actually peak somewhere between chlorophyll A and B, and then it dipped off, like all of these do, and then had a peak again where chlorophyll A is. So here, this would be our spinach, in black, and in red. Not only do we have carotenoids, but this is where we would see our carrot extract. Awesome. Okay, good. Questions, concerns? So that was the first experiment. The final 2 experiments for photosynthesis are pretty easy and straightforward, so we're going to go through those. Okay? All right, so now that we've established that, we also want to separate these pigments. We want to separate these pigments, and to do that, we're going to perform a particular technique on them. This technique is commonly used in a variety of different ways to separate things based on different physical or chemical characteristics. So I'm going to rewrite my pigments here. And I'm going to go chlorophyll B first. And I'll explain why here in a second. Then chlorophyll A. Then xanthophyll. And finally, carotene. Now the way that I've drawn these now on the board, or written these on the board, is based on the relative level of polarity. That is how polar are each of these pigments. Chlorophyll B is the most polar. Carotene is the least polar. And if I go up or down, I'm going to see that chlorophyll A is less polar than B, but more polar than xanthophyll. And xanthophyll is less polar than A, but more polar than carotene. So, what you then did with this information was you performed what's known as chromatography. Chromatography is a technique that separates molecules based either on their relative size or weight, or some chemical characteristic. And the chemical characteristic we're going to focus on here is polarity. So what you did was you took your spinach leaf and you rubbed it on a piece of paper. Now we have to be elaborate here. This process is called chromatography, so we got to come up with a nifty name for this paper, so we'll call it chromatography paper. Okay? So you took your spinach leaf and you rubbed it on this piece of chromatography paper. So all 4 of your pigments are in that line that came from your spinach leaf. Then you put this paper into a test tube, and in that test tube you had a solution, a solvent. Now again, this is chromatography, so we've got to be elaborate in the way we name the solvent, so we'll call it chromatography solvent. Okay? So you place your paper into this chromatography solvent. Now, going back to diffusion, and many of you have probably had the same issue if over the last few weeks you made the mistake of accidentally stepping in a puddle. Okay? Your pant leg goes in that puddle, the water doesn't just stay down at the bottom. Where does it start to diffuse? Up the pant leg. What is this chromatography solvent going to do on this paper? It's going to diffuse up the paper. So this chromatography solvent starts to diffuse up the paper. Now something happens to the paper when the solvent hits it. When the chromatography solvent hits the paper becomes polar. Okay? Now think about that real quick. The paper becomes polar. What kinds of molecules do polar molecules like to interact with? Do they want to interact with nonpolar molecules or other polar molecules? Other polar molecules. Which of these molecules is going to have the highest level of binding with the paper? Chlorophyll B. Now, here's what I need you to understand, because a lot of students don't understand what's happening here. When the paper becomes polar, that's the same thing as like a walkway becoming metal. The more polar a pigment is, the stronger the magnets that pigment would have in it if the ground here in this analogy were metal. So, which of these pigments, if chlorophyll B has the strongest magnet in it and carotene has the weakest magnet in it, which of those pigments would be able to move across the floor with the least amount of resistance? Carotene. The same thing's happening here. Chlorophyll B is so polar that it moves a little up the paper and then it sticks to the paper again. And then it moves a little more and it sticks to the paper. While carotene, it might stick just a little bit, but not a whole lot. So it's going to move up the paper quite a bit. So what you're going to see at the end of this whole experiment is these 4 different pigments are going to separate based on their polarities. The least polar pigment, i.e. carotene, is going to wind up closest to where the solvent moved in the paper. So your carotene is going to be closer to the top. Under that carotene, you're going to have this kind of hazy yellow mass. It's going to be xanthophyll. And then you're going to have 2 waves of green, one of which I'll draw in blue because I don't have a different color for the marker. Okay, so this first wave of green, which pigment is that going to be? Chlorophyll A. And then finally, this blue one here is going to be chlorophyll B. Questions, concerns? Anything I can clarify on that? All right, so last experiment in photosynthesis, and then we'll hit biotechnology to finish this out. If we run over a little bit, that's fine. If you need to leave at exactly 6, you can go. I'll continue talking and finish out biotechnology before I adjourn this review session. So let's go ahead and talk about the last experiment that was performed in the photosynthesis unit. And this was measuring starch storage in plants. So, plants are going to perform photosynthesis, but they might not need to use all of the sugar that they produce right then and there. So if they have excess sugar, they have to do something with it. They need a place to store it. And so the way they're going to store that sugar is as starch. And essentially what's going to happen is if the plant ever gets in a scenario where there's no light and it still needs to perform respiration to stay alive, it can break down this starch into glucose and it can perform cellular respiration. So what we did here was we had two different scenarios with the same type of leaf. So in one scenario, we had the leaf exposed to light. So here's my leaf exposed to this light. And in the second scenario, you might have had a single leaf that was just divided in half where this was done. So whether you had two leaves or one, it doesn't matter. Either you can just assume that this was happening to one half of the leaf and then the other half of the leaf had this other thing happening. But the fact still remains, either two separate leaves or one leaf had these two scenarios. In the second scenario, we covered the leaf, effectively blocking the light from getting to it. We placed aluminum foil around it so it did not get any light. So this second one here, the leaf gets no light. Whereas in the first one, the leaf gets light. Now, I have a really dumb question, but please just placate me. Which of these two leaves currently is performing photosynthesis? The top one, right? Because it actually has access to light, which is one of the three components you need to perform photosynthesis. All right, good. So the next thing we did was we took these two leaves and each of them was boiled in ethanol. Now the reason why we boiled them in ethanol was to remove all of the pigments from the leaf. We're not concerned with the pigments anymore. We want the leaf to be white because it's going to be easier to stain. Okay, so we boiled the leaves. We got these white leaves which we rehydrated by placing in water. And now the question we want to know is, which of these leaves has starch? Now remember, starch is produced if the leaf is performing a superfluous amount of photosynthesis. Okay? Superfluous means an overabundance or an excessive. Okay? So, which of these two leaves is going to be performing enough photosynthesis so that it should have starch reserves? The one on top. Okay? And how are we going to test to see if this leaf actually has starch? We're going to add iodine. We add iodine to this leaf, and when we do, we should see this leaf turn black, which means it has starch. Down here with this leaf, we add iodine, just the same, but what color should we expect to see? Like a brownish red. So this leaf up here should have been black, signifying that we have starch. This leaf here should be brownish red. And that signifies that we don't have starch. Questions, concerns about this exercise? Anything I can clarify? Make sense? Okay. All right, so that brings us to the end of photosynthesis.